My name is Steve Long. I'm the director of the RIPE project. RIPE is about harnessing the sun to feed the world. So what do we mean by that? What we're doing is developing technologies that can be applied to seed for some of the poorest farmers in the world to make that seed more productive so that they can produce more food on the limited land that is available to them and hopefully do it more sustainably. So we're calling this RIPE 100, and I'm going to try and meet that level in explaining it to you. So what is the background to RIPE? I guess first of all, what about me? My PhD, as you can see in this picture in the middle here, was actually on measuring photosynthesis. I've been measuring photosynthesis and analyzing photosynthesis and trying to change it for most of my career. On the left, I'm teaching photosynthesis in India, and on the right, I am involved in trying to help plant one of our experiments and being taught there how to do it correctly. So when I was at high school, the first Green Revolution was going on. And it's hard to imagine today, but at that time, there were very serious famines in China, in India, in other parts of Africa, where people were literally dying of malnutrition. The first Green Revolution really solved that problem by producing, in particular, more productive wheat, more productive rice. And in fact, I was very lucky a few years ago to meet Longping Yuan, who has been breeding rice for the last 50 years and a major part of the Green Revolution in China. And you can see on the wall behind us is one of the rice cultivars he's bred, with the panicle stretching almost from the flag leaf right down to the ground. But what he's complaining to me about is he can make a large amount of seed, but there's not enough photosynthesis to fill all of those seeds. So what are we going to do about it as physiologists? Now, if we look at the current situation, going back to 1960, if you like, the beginning of the Green Revolution, we've seen almost a linear increase in the amount of rice farmers getting globally per hectare of land. So it's been rising continuously. But if we look forward to population growth as it's predicted over the rest of this century, and the fact that the world population is becoming urban, demanding more food, the United Nations Food and Agricultural Organization say we need to be increasing like this, not this. So how are we going to do that? Now, we can look at what happened during the Green Revolution. The yield of a crop at a given location is determined by 4 interception efficiencies. So if we defend the plant from pests and diseases, we have good agronomy, we provide the nitrogen and water the crop needs, then what yield depends on, i.e. what we call the maximum yield a crop can obtain at that location, or the genetic yield it's capable of, that is the product of how much sunlight is available over the growing season, multiplied by the interception efficiency, i.e. how much of that light does it capture, the conversion efficiency, how good is it at converting that intercepted light into biomass, and then finally, how good is it at partitioning that biomass into the part of the crop we care about, so the grain of rice, the seed of soybean, and so on. So before the Green Revolution, we could say, well, partitioning efficiency was about 30% of the maximum. After the Green Revolution, we've almost reached what we think is the theoretical maximum for partitioning efficiency in our major crops. We've also improved interception efficiency, i.e. the crop covers the ground more rapidly, it captures more of the available radiation. But what hasn't been improved is conversion efficiency. So we might argue this is, if you like, the remaining opportunity to increase yield potential in our major crops, because we've done very little to improve that. So what makes us think we could improve it? Well, if we look at the theory of photosynthesis, where we lose efficiency, could say that we ought to be able to convert 5% of the visible radiation, i.e. photosynthetically active, into biomass. But what we achieve in most crops is around about 1%, and the very best is about 2.5%. So there's a big gap there. So how might we improve that? So one way we've looked at this is to look at the whole photosynthetic process, which you can see here, each step of it, and then represent that in a mathematical model. So it's rather like trying to model a car production line and saying, okay, if we've got a model of the production line, put it on a computer, predict where we need to be placing our workers to get the maximum productivity. And so what we set about doing, and Xingwen Zhu, who's part of Wright, led a lot of this work in my laboratory in the early 2000s, was to describe each of these reactions as a differential equation, usually capturing Michaelis-Menten kinetics or other kinetics of these reactions. And then with that system of differential equations, numerically integrating it. Now that proved to be more difficult than we thought, and we were stuck. And then one day at the computer lab, we met somebody called Eric de Sturla, who was working on rockets. And he said, oh, I have exactly the same problem when I simulate balancing rocket motors. And this is how I solved it. So we used his algorithms and they work for us. So this is why we have a rocket scientist on the first publication on this work. But he did add that actually what you're working on is much more complicated than what I'm working on. So, if somebody says photosynthesis is not rocket science, it isn't, it's much harder than rocket science. But what this allowed us to do was then predict where the bottlenecks might be. And from that type of modeling, we came up with many predictions on where we might be able to improve photosynthesis. And luckily for us, the Gates Foundation started to read those papers. And then they said, well, bring your team along and tell us how you think you can do this. And really at the end of sort of a year of discussion, the Gates Foundation said, well, I guess in summary, well, we're willing to put our money where your mouth is for a while, and you show us that this can really be done. So that's what the Wright mission is about, trying to show that this could actually be done. So our mission, long term, is to end hunger worldwide by improving the efficiency of photosynthesis to increase crop production. So the long-term aim is to equip farmers with high-yielding crops, and the Gates Foundation has developed another arm that we call Gates Ag One, which is taking our technologies and then translating these into seed for farmers. And that arm of the Gates Foundation began just a few months ago. I guess I should also add what is not our mission. We're not about improving stress tolerance, related processes such as translocation, transpiration, sink strength, etc. Our mission, as given by the Gates Foundation, is to focus on how, from a pure perspective, can we improve photosynthetic efficiency. So where are we? So the headquarters of RIPE is at the University of Illinois, but we also have subcontractors in UC Berkeley, in Louisiana, in Lancaster, in Essex, in Shanghai, and in the Australian National University. The crops that we are aiming at long term are maize, cassava, cowpea, soybean, and rice. So RIPE is basically focusing on those 5 crops. So how do we work? Well, we use the modeling to predict what would be the best bets for intervention. Then we have 7 different pipelines, different ways in which we might modify photosynthesis. And we use a common test bed so that we can say, okay, which ones are working best, which ones are showing the most promise. That common test bed here is tobacco. Why do we use tobacco? Tobacco is very easy to transform genetically, and it produces a large amount of seed. So within 12 months, we can go from transformation to field trials. We can't do that with our food crops. They would require about 4 years for the same process under current technologies. So we want, if you like, an easy test bed that tells us this particular change is really worth taking to the crops that matter. So this basically illustrates our pipeline, mechanistic models integrated into crop canopies, saying, okay, where might we intervene? Testing that in tobacco, taking that to field trials, and if that works, then we go on to the crops which really matter. So you might view this like a funnel. We can do billions of permutations in mathematical modeling. So from those, we distill out a much smaller number that we can test in tobacco. If they work in the greenhouse, then they go to the field. So we're now down from our billions to hundreds. And then if those work, they will go on to the crops that we really care about. And then Gates Ag1 will then work on taking those technologies, multiplying up the seed, more extensive field trials, and deregulating those crops for use by farmers. So I now go through our different objectives. And the first one is the mathematical modeling. And I've already given you some illustration of that. But of course, one of the things we also have to do is not only look at the, if you like, the underlying gene networks and biochemistry, but also the crop canopy, because photosynthesis in a crop canopy is not just leaves in full sunlight, it's also these leaves in the shade underneath. And so the whole system has to be optimized. One of the things we've been looking at is fluctuating light in the crop canopy. So we can then predict this from models and then say, what are the changes we might make to speed up the response of photosynthesis to these rapid changes? And so we've been looking at that in our target crops, looking at how light might fluctuate and then how we could adapt to that. So RIPE Objective 2, so this is one of our 7 pipelines beyond the modeling. And what the modeling predicted was, if you could take these systems that cyanobacteria have based on the carboxysome. Carboxysome contains the rubisco of the cyanobacterium. What happens in the cyanobacterium is that all carbon dioxide is converted to bicarbonate, so that out here you have a high concentration of bicarbonate. And none of the enzyme carbonic anhydrase, which converts that bicarbonate into CO2. Inside the carboxysome is carbonic anhydrase, so that in here, the bicarbonate's converted to carbon dioxide. That gives a very high concentration of carbon dioxide, which allows rubisco to act at maximum capacity, rubisco being the primary carboxylase of photosynthesis. And mathematical modeling said, if we could put this system into the chloroplast, then we could get a 60% improvement in photosynthetic efficiency. Now, this is probably the most difficult task that RIPE has taken on, because although cyanobacteria are the ancestors of chloroplasts, as far as we know, chloroplasts have not had this system for probably over 1 billion years. So now what the team at the Australian National University has set about doing is putting this system back into the chloroplast. And at the center of this is the carboxysome. So a shell with plate proteins, vertices, inside rubisco and carbonic anhydrase. So that requires several proteins to assemble that. And probably the most remarkable thing that RIPE has perhaps done is to do just that. It's to put those carboxysomes back into the chloroplast. And here you can see the first time this was seen, they engineered those genes into the nucleus with transporters to get the proteins into the chloroplast. And here is the chloroplast. Here are those carboxysomes. And just to check that these are really like the carboxysomes from the donating organism, the cyanobacterium, here are the carboxysomes centrifuged down, and they appear almost identical from the ones in tobacco to the ones in the cyanobacterium. So that was a major breakthrough. There is still more to be put into the carboxysomes. Carbonic anhydrase has just been put into the group, and now they need to focus on putting in these pumps as well to get this system working. So RIPE Objective 3 concerns photorespiration. In all of our C3 crops, so soybean, cassava, cowpea, rice, and other C3 crops, a major loss in photosynthesis is due to the process of photorespiration. That is that rubisco can react with oxygen as well as carbon dioxide. And as much as 50% of carbon assimilated here can be lost through this process. When rubisco uses oxygen, it produces a 2-carbon compound called glycolate. That is metabolized through other components in the photosynthetic cell, the peroxisome, the mitochondrion, before it then provides glycerate back to the chloroplast that can go back into the C3 or Calvin-Benson cycle. This requires a great deal of energy and lowers the overall efficiency of photosynthesis. However, microorganisms have different ways of metabolizing glycolate. And what Don Ort's group that Run Objective 3 have done is to put some of those alternative pathways, so you can see them labeled 1, 2, and 3, all different pathways from microorganisms into the tobacco cell. And they've also downregulated the transporter, which takes glycolate out of the cell so that they can keep the glycolate within the, out of the chloroplast, keep the glycolate within the chloroplast. And this has been very successful. So here are some of their modified plants. And you can see the modified plants are actually larger than the unmodified ones. So that was exciting, but what we need to really show, I should say at this point, a number of times people have shown a manipulation of photosynthesis in the greenhouse or controlled environment does something. But of course, for agriculture, we need to know that this really works in a replicated field experiment. So they took this to a replicated field experiment, and here you can see blocks of their different transgenic plants. And the important thing here is to look at the biomass, seeing that that pathway 3 produced up to 25% more biomass in this field trial. So now, of course, that is going on to crops. We'll have a field trial with potatoes this year, but it's also progressing into rice, into cowpea. Objective 4 concerns improving rubisco. Rubisco is notorious. It's the most abundant protein on the planet, but it is a very slow enzyme. It also reacts with oxygen, producing photorespiration. So it's an obvious target for improvement. Now, quite a bit of engineering was attempted in the 80s and 90s, which wasn't very successful. But this objective, led by the University of Lancaster, has been looking at diversity within crop plants and actually shown that rubiscos from different crops have substantially different properties. And so what they've developed is a high throughput system for identifying variation within rubisco within a particular crop. And they're focusing on cowpea and soybean, and what they're identifying is variation there that can then be used in breeding to get better rubiscos into these crops. Objective 5 deals with the other side of this. So rubisco, of course, requires RUBP to react with carbon dioxide. So what Objective 5, led by the University of Essex, has been looking at is how might we accelerate regeneration of RUBP? This involves looking at the whole Calvin cycle and where in the Calvin cycle we could intervene. So from the modeling, we've predicted sites where we might get improvements. And indeed, what the Essex team has done is then put in here 2 different genes and then combine them. And if we look at the productivity from these field trials, and this paper is just accepted by Nature Plants, it shows a combination which is giving us more yield in trials in the field. So again, this is a combination which will then move on to the crops we really care about. RIPE Objective 6 is looking at mesophyll conductance. What is mesophyll conductance? It basically describes the limitations of getting carbon dioxide from the intercellular space to rubisco in the chloroplast. So it's a combination possibly of biochemistry, membrane resistances, diffusive resistances to getting to this site. And of course, diffusion from the substomatal cavity to the wall of the cell. Now, one way we could tackle this is to maybe improve the area of cell to chloroplast. And we're also looking, we've looked at transporters, we're looking at a number of things that may affect this. If we can improve mesophyll conductance, it's a win-win situation because we improve photosynthesis, but we don't affect diffusion out, we don't affect water vapor passage. So you get more photosynthesis without losing more water. So it's a very attractive target. I'm just going to talk about one that we've been working on. One of the things we explored was, what if we make more airspace in the leaf? And so what was done in this work was to decrease investment in mesophyll cells. So we used a gene that was known from Arabidopsis, lower cell density it's called, to decrease the number of cells within the leaf. That would allow air to diffuse more easily, provide more surface area, but of course there would be less leaf mass there. So we weren't sure what the result would be. But in fact, what we found was these are our leaves with less cells, more airspace. So this is leaf CO2 uptake versus CO2 concentration. And you can see they're slightly better than the wild type. But what you notice on the right-hand side is that the leaf mass area is about half what it is in the wild type, i.e. we managed to achieve about the same photosynthesis with only half the mass. So how is that possible? Well, one of the things we've done by reducing the number of cells is that we're allowing light to penetrate the leaf more evenly, so that if light hits the upper surface, in the wild type, most of it is absorbed by the chloroplasts in the uppermost layer, and far less light gets to these layers. In our case, much more light is getting to these lower layers, and you can see, if you look at the absorption profile, the light is absorbed far more rapidly with leaf depth in the wild type than in the transformed plant. The other thing we've changed, and we were also able to measure this, is that carbon dioxide can diffuse far more freely in this leaf than this. So this is also important because if we can get the same photosynthesis with less investment in the leaf, then that means more biomass is available for productivity, i.e., for example, the seed of soybean. And I'll show you why that's important from another experiment in a moment. So objective 7 is about optimizing canopies. If we look at many crop canopies, we actually find surprisingly that light distribution is not optimal according to modeling. What we see in the crop canopy is these upper leaves absorb most of the light, and these lower leaves are actually starved of light. So there are various ways in which we could get more light into the lower canopy. One is to make these leaves lighter in color, so they absorb less of the light, more gets through to the lower leaves. Another way is to make them more vertical, so that more light gets through. And RIPE is trying both of those approaches. Why, I mean, you might ask, well, why are most canopies like this? And we think this is evolutionary because, of course, the crop ancestors grew in the wild where they're an individual in evolution competing against everything around them. If they can get up above their competitors, absorb all the light up here, their competitors down here are starved. Even though they can't use that light, they prevent their competitors from getting it. Now, we have some evidence that this hypothesis is true, for example, in soybean, where our modeling said, well, the soybean canopy is producing too many leaves. It can't make use of all that light. So we could test this experimentally quite easily. And basically what we did was to cut off every third emerging leaf, i.e. the plant could then not invest in the growth of these leaves. And what that did was to increase seed yield by 8%. So that told us if we can find ways of investing less in leaves after canopy closure, then we could get more yield. So this is where what I showed you under mesophyll conductance could also be an important finding. Now, Don Ort's group is looking at ways of making the upper canopy lighter using an inducible promoter so that it only starts to get lighter once the canopy is closed. And because before closure, the leaves can make use of all the light they can get. And you can see that is showing some promising results. Objective 8 is the final of these 7 pipelines. So I showed you in the modeling that if we actually look in the real world, much of crop photosynthesis is occurring under fluctuating conditions of light. When we examine photosynthesis in fluctuating light, its efficiency is much lower than at steady state. Now, one of the ways in which it's inefficient is that when a leaf goes from sun to shade, and this is occurring all the time over the course of a day, because as the sun moves across the sky, these leaves are going in and out of shade just due to sun canopy geometry. And at the level of a chloroplast, this is an instantaneous change, but it takes many minutes for that chloroplast to readjust to the now shade conditions and, if you like, ramp its photosynthesis back up to the highest level it can achieve in the shade. So in this project, which is joint between my laboratory and Chris Niogi's in California and now Vanna Kromdijk's in Cambridge, what we reasoned from modeling and Chris's work with Arabidopsis was that we needed to upregulate 3 genes controlling this rate of recovery. And so that is what we did. And in the greenhouse, we could see that independent transgenics carrying this 3-gene cassette appeared more productive. We then went to field trials, and with these replicated field trials, we then actually saw that the 3 events we took to the field all showed substantial improvements in productivity. And this trait is now being transferred to cowpea, soybean, and rice. And so advancing translation is about, once it's been validated in tobacco, to then take it to the crops that we actually care about. Now, we also need technologies. As we get more and more plants in the field, we need technologies which are much more high throughput than those we've been using. And so another branch of RIPE, in this case headed by Carbonaci's lab, is looking at hyperspectral measurements and reflectance, other reflectance measurements that might allow us to screen many more plants more rapidly in the field to say, Are they really showing the phenotype we're using? And can we find natural variation that we could exploit? As I mentioned, we're also now taking traits to the crops we care about. And this is a data from Stephen Burgess on cowpea that's been transformed by TJ Higgins' group, where we're then looking here to see expression of the 3 genes we actually care about. And here you can see these were different events planted out in the field in Puerto Rico, where we have a field site to advanced translation where this work was done. So finally, sort of to just give you a RIPE facilities update, one of the things we've realized is that as we get more and more events, we can't screen these easily at the numbers we're producing. So we had over 1,000 individual events last year. To actually screen that requires an army. So we now need automated phenotyping where we can monitor growth, photosynthesis, water use throughout the growth of the plants, not just simply higher biomass. And so with the supplementary funding that the RIPE Foundation provided to advance translation, we're now building out a RIPE facility which will provide that high throughput phenotyping for the whole of RIPE. And so this is an idea of what that will look like. There'll be an overhead gantry that will be scanning our plants as they develop. They're on load cells so that we can determine how much water they use each day, how much weight they put on each day. We also have to ramp up transformation, and we're now moving not only to ramping up our tobacco transformation for the whole of RIPE, but also putting traits directly into soybean. And so opposite our high throughput phenotyping facility is a high throughput transformation facility. And so we're now going to have many more laminar flow hoods in there, and all of the steps will be integrated into a flow path for increasing our transformation capacity. When we get to the field, again, we need to increase transformation capacity there. And this is being done by a spider cam, which should be installed in the field this fall. So, of course, RIPE wouldn't be RIPE without our team. And we're very lucky to be involving most of the top photosynthesis laboratories in the world. So, and our funding, of course, is from the Bill and Melinda Gates Foundation, the Foundation for Food and Agricultural Research, and UKA. And they've been fantastic funders, have been engaged in all of our work. And so, you know, we're extremely grateful for them to taking a risk on us, which I think nobody else would have taken when this started. And RIPE would not be RIPE without all of you. So thank you.